With that, I'd like to invite Eric to the podium. We will all congratulate him and hear his lecture as the Richard L. Schilsky Award winner for 2011.
Monica, Dr. Bertagnoli, distinguished guests, and colleagues, I am hugely honored to be the recipient of an award bearing Rich Schilsky's name. You know, I entered oncology to contribute, and what a huge contribution Rich has made. If I can contribute but a fraction of what Rich has, I would consider myself hugely, hugely successful. So, what do you do if you're Richard Schilsky and have already accomplished it all? You've been CALGB Group Chair, you've been ASCO President, member and Chair of ODAC, a Cancer Research Center Director, a Section Chief, Associate Dean, member and Chair of the NCI Board of Scientific Advisors, and I think most tellingly, a member of Scientific Advisory Board to over 20 cancer centers, including mine, for which my institution is deeply indebted. What do you do if you've already done that? Well, the answer to me came in a flash. You replace Letterman. And in that vein, I'd like to talk to you about Schilsky's top 10 reasons to participate in the CALGB. In order to set the stage for that, permit me to provide a brief history of the CALGB GU Committee. As Monica alluded to, the origins of the GU Committee lie in the GU Working Group constituted by Emil Frey in 1987. Under the leadership of Nick Vogelsang, the Working Group contributed to intergroup efforts and launched its first trials in advanced prostate cancer using high-tech agents as high-dose megase and mitoxantrone prednisone, CALGB 9181 and 9182. In 1993, the CALGB Board of Directors felt that the working group had sufficient legs to be a full committee, but it was constituted as a more focused prostate cancer committee. By the mid-90s, the prostate committee had developed a correlative science working group and had launched pivotal studies, mostly in advanced prostate cancer. In late 1999, Nick Vogelsang stepped down as chair, and at around the same time, the prostate committee became the GU committee and was charged by Dr. Schilsky to expand its scope to all of the major urologic malignancies and to further expand its early stage disease portfolio. So, within this context, I'd like to share with you what I believe are the top 10 reasons to participate in the CALGB, and I hope that junior members in the audience pay close attention. Number 10, get mentored. Nick Vogelsang chaired the GU Working Group, and when it became a prostate committee, he was joined by Nancy Dawson as vice chair. In addition to launching the group, Nick and Nancy's accomplishments are the mentoring of a large number of fresh young things, myself included. I still remember my first GU Working Group, just a year out of fellowship, and sitting in a room with luminaries like Vogelsang, Dawson, Gelman, Kantoff, Scher, Trump, Raghavan, that were not only incredibly accomplished, but incredibly opinionated and incredibly loud. After I ventured a timid little comment, and for those of you who know me, I can on occasion be timid, and then withstood an absolute barrage of withering comments, Nick smiled his famous toothy grin at me and said, "Not a bad thought for west of the Mississippi." The opportunity to offer support to the next generation in similar ways is wonderful. Number 9, you get to work with the leaders in the field, the best of the best. As the prostate committee broadened its scope, I had the opportunity to help constitute the next group of cadre leaders with an absolutely astonishing group of talented individuals who I would like to honor. Anthony D'Amico led our radiation oncology cadre, while Jim Moeller led our urology cadre. Jim has also served as vice chair of the committee. Together, Anthony and Jim brought incredible depth and experience to our localized prostate cancer efforts. My friend, Kevin Kelly, led the advanced prostate cadre. Dean Bajoran, the bladder cadre, Walt Stadler, the renal cadre, while Phil Kantoff brought incredible strengths as leader of our correlative science group. More importantly, I appointed Phil, and he carried with great style and flamboyance the title of Director of Dining and Entertainment, responsible for selecting the restaurant we would all go to after our meetings. I should point out that we're currently recruiting for this critical position, so if you're interested, see me after dinner tonight. Not only are these truly accomplished and committed cancer investigators that I've been privileged to work with and learn from, but they've become valued and dear friends. And without question, there's huge gratification in helping to develop the next generation of leaders in the committee. In March of 2010, a new group of cadre leaders was appointed to lead efforts with the GU committee. Jim Moeller, as vice chair, and I have stayed on to provide some gray hair continuity. Michael Morris assumed leadership of the advanced prostate group. Mark Hurwitz leads the radiation oncology efforts. Jonathan Rosenberg leads the bladder cancer group, and Dan George the renal cell group. Phil Febo also serves as vice chair for the committee and is charged with our correlative science group. Each one of these talented individuals brings tremendous skills and expertise, and I think spells nothing but good things for the future for the committee. Without question, one of the greatest privileges of working in the CALGB is witnessing and participating in the sweep of oncologic history. And I'd like to just give you a sense of that. This is what's happened in prostate cancer during the timeline that I've shown you. We've gone from one FDA-approved agent, mitoxantrone, to now look at all these agents. And those of us that have been privileged to be in the field in this area have seen an absolute transformation of the care that our prostate cancer patients get. The same thing holds true for renal cell carcinoma. Between 1992, when IL-2 was approved, and December of 2005, when sorafenib was approved, there was a virtual desert. And now, look at all these agents. Again, the CALGB GU committee has played a part in these, and I'll show you some of it. Our next challenge is to have the exact same kind of progress in remaining malignancies, including bladder cancer. So as a corollary, without question, reason number 6 for me of the top 10 reasons to participate in the CALGB is to contribute on the war on cancer. The prostate cancer landscape in 1987, when the GU working group was established, was as follows. PSA was new, and most physicians were not using it. You see here on this graph of prostate cancer incidence that we were just at the beginning of the uptick attributed to PSA screening. Prostate cancer was diagnosed in 70,000 men per year in the US. It was metastatic in nearly 40 to 50% of patients at presentation. It currently is metastatic in about 15% of patients. The median survival of hormone refractory disease, which at the time we called androgen-independent prostate cancer, was 9 months. We're currently hovering at 24 months. Chemotherapy use was negligible, and secondary hormones, while there was some use, was not based on phase 3 trials or evidence-based medicine. So here are our contributions as a CALGB. I already mentioned to you CALGB 9181, this trial of Megace. And these are CALGB's contributions with regards to the role of secondary hormones in castrate-resistant prostate cancer. We conducted the first trials with secondary hormones. We helped define the androgen-independent prostate cancer state. The CALGB conducted the very first prospective study of anti-androgen withdrawal and subsequent secondary manipulations, in this case, ketoconazole. We demonstrated that the mechanism of resistance previously attributed to androgen receptor mutations was in fact incorrect, and began to set the stage for our understanding of AR amplification as one of the most important targets in advanced prostate cancer. This went in hand with the observation that adrenal androgens are important, and that if they are ablated, that progress can be made. This in turn led to direct work done leading to the approval of abiraterone acetate for prostate cancer. We contributed as well to the role of chemotherapy in castrate-resistant prostate cancer. CALGB conducted the first phase 3 trial of mitoxantrone together with an NCIC Canada trial that led to FDA approval of this drug the first cytotoxic approved for chemotherapy. We were the first multi-center organization to conduct a trial of docetaxel, CALGB 9780. Around that time, and I'll comment on this later, we demonstrated the prognostic role of VEGF levels in prostate cancer and developed phase 2 and 3 trials targeting bevacizumab, VEGF with bevacizumab. And finally, under the leadership of Susan Hollaby, we developed a widely used prognostic model for castrate-resistant prostate cancer. With regards to kidney cancer, in 1999, the landscape was, as I told you, IL-2 was the only approved drug. Phase 2 trials in metastatic disease were quite challenging because of this phenomena that many patients develop stable disease, and it wasn't clear if that stability was due to the drug or due to the natural history of the disease. The role of angiogenesis was just beginning to be appreciated. There were no TKIs on the horizon, and I'll remind you that bevacizumab was first approved in 2004. So what did the CALGB contribute? Well, under the leadership of Mark Ratain, Drs. Rosner and Stadler conducted the first contemporary randomized discontinuation trial in oncology. We all think of these as sort of, well, they're pretty standard. But in fact, this is an example of how CALGB contributes not only to the discovery of novel agents, but to the discovery and development of methodology. CALGB undertook the first contemporary anti-angiogenesis trial in renal cell carcinoma with CALGB 90206, interferon versus interferon bevacizumab, which together with a European trial led to the FDA approval of bevacizumab for the treatment of metastatic renal cell carcinoma. And along the way, we did smaller phase 2 trials that demonstrated little benefit to two approaches that had achieved great favor, a gemcitabine-capecitabine combination and a non-myeloablative marrow transplant. The fifth top 10 reason to participate in the CALGB, without question, is surrounding oneself with talented and dedicated colleagues. John Taylor, the GU Committee's protocol editor, and Susan Hollaby, the GU Committee statistician, are the most professional, hardworking, and dedicated team players I know. John and Susan, your commitment to excellence has kept the committee on an even keel, even when your committee chair has slipped up. Working with people of this caliber is an extraordinary privilege. In a certain sense, John and Susan are iconic for the amazing, truly remarkable CALGB staff and faculty in Chicago, Boston, and Durham, who I would like to salute at this point and thank. Reason number 4. Working in the CALGB teaches you how to play in the sandbox. Sometimes we take it for granted that multidisciplinary research just happens, but we all know that it takes hard work. At the CALGB, there is a culture of collaboration and cross-disciplinary work, which I suspect is a role model for most of our home institutions. As an example, CALGB 90203, chaired by James Eastham, a urologist, and co-chaired by Paul Monk, a medical oncologist, is one such example. This is a tough accrual, randomizing patients who are to have a radical prostatectomy to receive neoadjuvant chemohormonal therapy or not. We have accrued 348 of 750 patients, and we're going strong.